Good morning. My name is Mario Lloret Torres, and I'm a graduate student at the University of Puerto Rico Medical Sciences campus at the Department of Anatomy and Neurobiology in the lab of Dr. Jennifer L. Barreto Estrada, and today I will be presenting this project titled Hippocampal, Amygdalar and Accumbal BDNF expression in morphine extinction. So in our lab, we study addiction, which is a chronic relapsing disease, which consists of three major phases. The first is a binge intoxication phase, where the subject takes the drug. This is followed by the withdrawal phase, in which the subject suffers negative effects caused by not taking the drug. And finally, the preoccupation anticipation phase, where the subject seeks out the drug to alleviate their aversive effects. So studying addiction is important because it is a global public health crisis. It's even worse in the United States, where 67,367 people died of opioid-related overdoses just in 2018. In the United States, opioid-related deaths have been increasing exponentially over the last few years. Currently, addiction is treated with a combination of medication-assisted treatment, which is what's commonly known as methadone clinics, although other alternative drugs are used, and also behavioral therapies in tandem. However, a large subset of patients is resistant to this type of treatment and could ultimately end up dying from an overdose-related death. So one behavioral therapy that's used for addiction is exposure-based therapy, which is when you take an addicted patient who is used to taking a drug in a particular setting, and then you expose them to that same setting in the absence of the drug to try to dissociate the drug from that setting. So this has been replicated in animal models using these types of two-sided chambers. It's essentially a box with two different color sides. And then the animal is exposed to drug on one side of the chamber, but not the other. You can then allow the animal to explore the complete box, and you will see how its preference for the drug-associated chamber will increase. So on the right, what you have is an example of what you would expect to see from this type of experiment. Animals would start with no real preference for either side, and then after being exposed to drug for several days, they would develop a preference for the drug-paired side. However, if they're exposed to the full box without the drug for several more days, they lose their created preference and return to having no preference for either side. But how do these changes in behavior relate to brain activity? Well, we know that addiction is associated to a specific subset of brain regions known as the reward circuit, and this includes the VTA, the hippocampus, the prefrontal cortex, as well as the amygdala. Extinction, which is the part where the animal learns that the context is no longer associated with the drug, is said to be regulated heavily by this area here, which is called the ventral striatum nucleus accumbens. Now, the ventral striatum nucleus accumbens is said to regulate extinction through its glutamate signaling. And you can see it here receiving lots of glutamate signaling, that's the blue arrows, from the other areas of the reward circuit. So while we have a pretty good idea that the nucleus accumbens is regulating extinction, what we don't yet know is what is happening at the molecular level. Originally, we set out to identify genes associated with morphine extinction. Because the nucleus accumbens was so extensively associated with extinction, we created a gene plasticity profile of the nucleus accumbens from rats that underwent extinction training. So to look at extinction, what we did was that we conditioned our animals using our two-sided chamber, and then we tested to see if their preference for the drug-paired side had increased. And then we put them through extinction training where they were exposed to this drug-paired side in the absence of drug for several days so that we could finally see if their conditioned preference had been erased. What we found was that there were actually two distinct phenotypes. This orange line here represents our control. These are animals that were conditioned but did not receive extinction training. And what you see is that they start out here low on baseline and then they're conditioned. So their preference for the drug paired side increases, but because they're never put through extinction training, the preference for the drug paired side never really goes away. The blue line is the animals you would expect to see. They start out low, then they're conditioned to prefer the drug paired side, and then after extinction training, you can see that they extinguished their learned preference. But we also found a third phenotype, animals that started low and then conditioned for morphine, but even after being put through extinction training, did not extinguish their previous behavior. What you see down here is just the average values for all of the animals. We decided to look at these groups individually to see if we could find any genetic differences between the two. We then extracted the nucleus accumbens from the brains and ran an RT2 profiler PCR array for synaptic plasticity to see if there were any genetic differences between the groups. We found several differentially regulated genes in the groups, but perhaps the most statistically significant was brain-derived neurotrophic factor, or BDNF. BDNF mRNA was found increased in animals who successfully extinguished for morphine, but not in animals who failed to do so. And this led us to believe that BDNF could be playing a major role in extinction. BDNF is a growth factor that has long been known to play a major role in extinction of fear, and more recently in extinction of addictive behaviors. Because our increased BDNF was in the group of animals who successfully extinguished, we think it could be playing a major role. However, we had no way of knowing whether this increase in mRNA was actually translating into active protein. So in this project, we wanted to determine the levels of BDNF protein expression in the nucleus accumbens, as well as other reward circuit related regions, like the hippocampus and amygdala, of animals who underwent extinction of morphine conditioning. We hypothesized that animals who successfully extinguish morphine will have increased levels of BDNF protein expression in these regions. What we did was that we took a separate group of animals and conditioned them exactly as we'd done before, and then we extracted the reward circuit related regions from their brains, but instead of running real-time PCR, we used Western blots to determine protein expression levels. And here's what we found. This is just a reminder that there are three groups: those who extinguished the way we expected them to, those who failed to extinguish, and the control group. And on the right, what we have is the result from the Western blot. Here are some representative bands so that you can see what it looked like for those groups. And what we saw was a trend that showed more BDNF in the animals who successfully extinguished, and not the ones who failed to extinguish, nor the controls. However, none of this has yet to reach statistical significance. When we looked at the hippocampus, a region with much higher expression of BDNF, we found a significant increase in the animals who successfully extinguished morphine compared to the controls that was not observed in the animals that failed to extinguish. This is very similar from our findings in the gene profile. Oh, this was done with a one-way ANOVA. So we actually wanted to see if the expression levels for BDNF in these animals were in any way related to their behavior. So we ran a linear correlation including both groups of animals, those who failed to extinguish, which are in purple, and those who extinguished, which are in blue, and found that it was correlated. Finally, when we looked at the amygdala, which is more related to emotional responses, we can see a trend in both extinction animals and animals who failed to extinguish to have more BDNF, but not significantly more as of yet. This could mean that at least in the amygdala, the increase is more related to extinction training than to the actual extinction process. So in summary, because extinction is used in behavioral therapies for addiction, it's important that we understand the molecular mechanisms that drive the. That is why we created a transcript profile to identify extinction-related genes. We found an increase in BDNF, but we had no way of knowing whether this increase was translating into the protein level. We used Western blots to try to validate this, and we found an increase in BDNF in the hippocampus. And we found that this increase in expression also correlated with those animals' behavior. We believe BDNF could play a major role in extinction, as confirmed by its increased expression in the nucleus accumbens, hippocampus, and amygdala. Because of the similarities between extinction and preclinical therapies, we think BDNF could be a potential target for future addiction therapies. So for future studies, we would like to increase the sample size of our Western blots, as well as measure BDNF in the prefrontal cortex, which is another region associated to extinction. We also want to determine BDNF concentrations in animals who've been exposed to low-frequency deep brain stimulation, as we have some data that could suggest this is therapeutic. Finally, we would like to pharmacologically target BDNF in the regions of the reward circuit. I would like to thank my lab, my PI, Dr. Jennifer Barreto Estrada, our lab technician, Roxana Ayala, who did a lot of the work on the Western blots I showed you today, our undergrads, Pedro and Lisman, Fredison Martinez Rivera, who is a former graduate student and who is responsible for the gene profile experiment I presented today, the MBRS RISE program, as well as the MBRS SCORE program, and Dr. Gregory Quirk's lab for always being available to us, especially Sarkali and Hector, as well as the Center for Collaborative Research in Health Disparities.